Okay, and on to our third and final set of notes on biochemistry. In this, we'll discuss proteins and nucleic acids. So proteins are the polymers of amino acids. They contain carbon, hydrogen, oxygen, nitrogen, and often sulfur. Proteins are the workhorse of the biochemical world. They basically do everything, to the point that actually for a very long time, we believed that genetic material was passed down through proteins because bluntly, they just did everything else, so why not? Okay, so they express genetic information. That's not passing it on. Keep in mind that nucleic acids actually code for proteins. Okay, so they're the expression of genetic information. They are a structural material. They provide energy. A little bit longer term energy than carbohydrates. For example, if any of you guys know somebody who's diabetic, if they eat a lot of meat, often they'll spike a high later on during the day. They are enzymes. They are membrane proteins. You have receptors, membrane transport, antigens. They're involved in transportation, such as hemoglobin. They're storage, and they make antibodies, muscles, and a whole litany of other things. Okay, so there are 20 common amino acids, but they all have the same basic structure. I've got an amino group. I've got a carboxyl group. I've got a carbon in the center, and then their side chain varies. And we simply refer to it as R. R is just the general generic term. And then whatever, the simplest of which is just a hydrogen, and the most complicated of which can become a long chain. And they vary in size, electrical charge, and solubility. These side chains are what make amino acids, each particular amino acid, either hydrophilic or hydrophobic, which then affects protein 3D structure. They are built via dehydration synthesis. This is just a nice little thing. Again, I mentioned it when we first discussed dehydration synthesis versus hydrolysis. And the types of bond that forms are peptide bonds. That's what we refer to them as. The sequence of amino acids determines the function of the resulting protein. So to begin with, you have a dipeptide, which is 2 amino acids, and we get a polypeptide, and then a protein, which is 50 plus amino acids and or 2 polypeptide chains. And we'll talk about what that means in a minute. Protein structure. You have to know the 4 levels of structure, of protein structure. I will be asking you questions about it. This is another one of those things like the functional groups. It'll be on this test. It'll be on your midterm. It'll be on your final. If you take other biology classes, it will be in it. So you have to know the 4 structural levels. We have primary, secondary, tertiary, and quaternary. I know they did such a lovely job of naming them so uniquely. 1, 2, 3, and 4. So primary structure, this is simply the chain of amino acids. This is, does it go arginine, tryptophan, tyrosine, or does it go arginine, tyrosine, tryptophan? This is all that it is. It is just simply going through and reading the letters out loud. Okay, that's basically what it is. There's no folding, there's nothing it's just the amino acid sequence. Secondary structure, we get some folding. We get alpha helices and beta sheets, which are due to your bonding with R groups. Basically, if you see the word alpha helix or you see the word beta sheet, you know that we are talking about secondary structure. Tertiary structure, this is when it folds in on itself. This is going to be a 3D structure. I like it because tertiary means 3, and in this case, I'm talking about a 3D structure. Okay, so, and then this is where we get attractions form between different alpha helices and beta sheets. You get folding and twisting due to hydrogen interactions, ionic interactions, covalent. You get disulfide bonds between the different R groups. And then quaternary structure. This is when I have more than 1 amino acid chain. Okay, so for example, each chain has primary, secondary, and tertiary structure, but you only, when you add more than 1 chain together, do you get quaternary structure. And our fabulous example of quaternary structure is always hemoglobin, because you have a total of 4 subunits in hemoglobin, which makes it a little bit easier to remember quaternary structure. If I heat it up too much or I alter the pH too much, then I disrupt my bonding and a protein denatures, which means it unwinds to its primary structure. No more alpha helices and beta sheets. It's just sort of all loosey-goosey. Luckily, most of the time, if we put proteins back into ideal circumstances, they will renature, okay? They will fold up again. It's often not a permanent change. But this is what, like somebody having a fever for a long period of time, denaturation will happen. And actually, one of the reasons is our body's reaction to an infection is because it does help kill the invader. Okay. And last but not least, nucleic acids, personal favorite of mine. Okay, these are composed of nucleotides. So this cute little circle, hold on here, circle, pentagon, rectangle thing, anytime you see that, that means we're looking at a nucleic acid. That's sort of the general depiction of one, okay? They store genetic information. They're important for intracellular messages and short-term energy transporter. ATP, which is adenosine triphosphate, is our go-to, like, this is what cellular energy is. When we talk about cellular respiration, we talk about it in forms of, in the number of ATP produced, okay? That is what we're talking about. So that's nucleotide is one of those. So I have nucleic acids. I have DNA, right, which is our genetic blueprint. It's a double-stranded helix. My adenine binds with my thymine, my guanine to my cytosine. We'll talk about that more later when we talk about replication, but the straight letter goes together and the curvy letters go together. Okay, and then I have RNA, which is involved in protein synthesis. It can leave the nucleus, whereas DNA cannot. It is a single strand, and I now have uracil instead of thymine because our body needs to know it can leave the nucleus. It's sort of like speaking in a different, same language, different dialect. Again, more detail when we get to transcription and translation. So adenosine phosphates, I have adenosine monophosphate, adenosine diphosphate, and adenosine triphosphate. They carry energy. So what happens here, this is ATP. When I remove this phosphate, a whole bunch of energy is released, okay? And then I can put it back on. ATP will get recycled back to ADP, back and forth. Again, when we get into respiration and whatnot, we'll talk about that. Nucleotide coenzymes carry hydrogen and electron. This is NAD and FAD in photosynthesis and respiration. And you will never, ever, ever have to know what NAD and FAD stand for. You're welcome. Cheat sheet, DNA versus RNA. DNA is double-stranded versus RNA is a single strand. DNA contains thymine, whereas RNA contains uracil. DNA is deoxyribose sugar. RNA is just a ribose sugar. That's why you get the different letters at the front, okay? DNA is long-term storage of genetic info. RNA is used in protein synthesis. DNA is only in the nucleus, whereas RNA can leave and go to the cytoplasm. DNA is very highly susceptible to ultraviolet, damaged by ultraviolet rays, whereas RNA is much less likely to be damaged by it, partly because it lasts for a considerably shorter period of time in the cell. And that, ladies and gentlemen, is the extent of your notes on biochemistry. Good luck with it all.